Welcome to G Fun Facts Online. For centuries, our understanding of the deep past came from silent clues, stone tools, fossilized bones, and paintings on cave walls. Then came the study of ancient DNA, which gave these ancestors a voice. But DNA is fragile. It's a delicate blueprint that fades with time, especially in the warm climates where so much of human history unfolded. What if there was another record? A tougher, more resilient storybook of the past. One that could survive not just for thousands, but for millions of years? There is. And it's hidden inside the very building blocks of life, ancient proteins. This is the world of paleoproteomics, a revolutionary field that is allowing us to read the lost chapters of our history. By analyzing these incredibly durable molecules, scientists are identifying mysterious human ancestors, figuring out what was for dinner 8,000 years ago, and pushing the timeline of our own evolution back to a period we once thought was lost to time forever. So why are proteins the new superstars of archaeology? While DNA holds the instructions, proteins are the machines and structures that actually do the work in a living organism. They are the bricks and mortar. And just like bricks can survive long after the architectural plans have turned to dust, proteins can persist for an astonishingly long time. They get locked inside the mineral matrix of bones and teeth, shielded from decay for hundreds of thousands, or even millions of years. This incredible longevity makes them a treasure trove of information. The most common source is bone and teeth. The hard, mineralized structure acts like a natural time capsule. Collagen, the main protein in bone, is especially sturdy and has become the workhorse of the field. And tooth enamel, the hardest substance in your body, is an even better protector, preserving proteins that are rewriting our understanding of human evolution. But it's not just skeletons. Scientists have pulled proteins from the shells of million-year-old ostrich eggs, giving us clues about ancient birds and their environment. And perhaps most excitingly, they're looking at the artifacts our ancestors left behind. Imagine an ancient clay pot. Microscopic residues of food can get trapped in its porous surface. By analyzing these residues, we get direct evidence of what people were cooking and eating. We can even find protein traces on stone tools, revealing exactly which animals were being hunted and butchered. In very rare cases, like bodies preserved in permafrost or arid deserts, even soft tissues like hair and skin can hold on to their protein secrets, offering a truly unparalleled glimpse into the biology of an ancient individual. So we know where to find these molecular treasures, but how do scientists actually read them? The process is meticulous. First, they have to free the proteins. For a bone or tooth, this means using a weak acid to gently dissolve the mineral matrix, releasing the proteins that were trapped inside. For an artifact, like a piece of pottery, they use special chemical solutions to carefully lift the protein residues off the surface without destroying them. Once the proteins are extracted, they're often too big and complex to analyze directly. So, scientists use enzymes to act like molecular scissors, snipping the long protein chains into smaller, more manageable pieces called peptides. Now comes the high-tech part, deciphering the molecular code using a machine called a mass spectrometer. There are two main techniques. The first is a rapid screening tool called ZoomS, which stands for Zooarchaeology by Mass Spectrometry. Think of it like a molecular barcode scanner. ZoomS looks at the collagen peptides and creates a unique peptide mass fingerprint. This fingerprint is different for different species. So if an archaeologist finds a pile of tiny, unidentifiable bone fragments, they can use zooms to quickly sort them. Is this piece from a sheep, a deer, a pig, or something much more exciting, like an ancient human? It's a fast, cheap way to analyze thousands of bones, and it has completely transformed archaeology. But what if you need more detail? That's when scientists turn to a more powerful technique, often called shotgun proteomics. This method doesn't just create a barcode, it reads the entire book. It separates the complex mixture of peptides and then analyzes them in two stages. The first stage measures the mass of the peptides. The second stage actually breaks the peptides apart and measures the mass of the fragments. This fragmentation pattern reveals the exact sequence of amino acids, the letters that make up the protein word. By searching this sequence against massive databases of known proteins, scientists can identify the specific protein and the exact species it came from. It's this deep analysis that is leading to some of the most groundbreaking discoveries about our own past. And the stories these molecules are telling are nothing short of extraordinary. For a long time, the reach of ancient DNA was limited to a few 100,000 years at best. Paleoproteomics has shattered that limit. In 2020, a team of scientists did something incredible. They analyzed the tooth enamel from an 800,000-year-old fossil found in Spain, belonging to a species called Homo antecessor. They successfully sequenced proteins from it. The analysis revealed that Homo antecessor wasn't a direct ancestor, but a close sister lineage to the group that includes us, Neanderthals, and another mysterious group of ancient humans, the Denisovans. This placed a crucial new branch on our family tree from a time period that DNA could never reach. Speaking of the Denisovans, they were an enigma for years, known only from a few tiny bone fragments and teeth found in a single cave in Siberia. Then, in 2019, a jawbone discovered in a cave high on the Tibetan plateau was analyzed. There was no DNA left, but scientists were able to extract collagen proteins. The protein fingerprint was a perfect match for the Denisovans. This was the first time an ancient human fossil had been identified solely by its proteins. It proved that Denisovans were not just confined to Siberia, but were adapted to the harsh, high-altitude environment of Tibet over 160,000 years ago. The story gets even older. In another landmark study, researchers managed to recover protein fragments from the 1.77 million-year-old teeth of a Homo erectus fossil from Dmanisi in Georgia. These proteins were incredibly degraded, but their successful recovery proved that paleoproteomics has the potential to probe the very dawn of our genus, Homo, a time nearly 2 million years in the past. But it's not just about drawing our family tree. Ancient proteins are also giving us an intimate look at the daily lives of our ancestors, starting with what they ate. At a famous Neolithic site in Turkey called Çatalhöyük, archaeologists had been finding strange, calcified deposits on the inside of 8,000-year-old pottery. For years, they weren't sure what it was. A team decided to analyze these crusty bits for proteins. The results were astonishing. They found proteins from wheat, barley, and peas, but they also found milk and blood proteins from animals like cows, sheep, and goats. They could even distinguish between different types of milk proteins, which strongly suggests that these early farmers weren't just drinking milk, but were already processing it into products like yogurt or cheese 8,000 years ago. The evidence was right there, stuck to their dishes. We also have the famous case of Otzi the Iceman, the 300-year-old mummy found preserved in the Alps. Protein analysis of his stomach contents gave us the menu of his last meal: ibex and red deer meat, eaten with einkorn wheat. The analysis was so detailed, it could even identify the specific muscle tissues head consumed. By looking at protein residues on stone tools, we can even see what was on the menu 250,000 years ago. One study found traces of horse, rhino, and even duck on tools from a desert oasis, painting a vivid picture of the diet of early hominins. This new science is also opening a window into the health and diseases of the past. Proteins from pathogens, the germs that make us sick, can sometimes be preserved in ancient remains. For example, researchers have found proteins from Mycobacterium tuberculosis, the bacterium that causes TB, in the bones of ancient people, giving us direct evidence of the disease's long history with humanity. By analyzing our ancestors' immune-related proteins, We can even see how their bodies responded to infections, giving us insights into the evolution of our own immune system. Of course, the field isn't without its challenges. Time is the ultimate enemy. Even though proteins are tough, they still degrade over millions of years, becoming fragmented and harder to read. But the biggest challenge is contamination. A single skin cell or a stray bit of dust from the lab can introduce modern proteins that can swamp the ancient signal. Scientists have to work in ultra-clean labs and take extreme precautions to distinguish the ancient molecules from modern intruders. And finally, their ability to identify a protein depends on having something to compare it to in their databases. If the species is extinct and its protein sequences are unknown, identification can be difficult. Despite these hurdles, the future of paleoproteomics is incredibly bright. The technology is getting better all the time. New mass spectrometers are more sensitive, allowing scientists to analyze even smaller and more degraded samples. They are working on ways to expand their databases and are even pursuing non-destructive methods, so they can analyze precious fossils without having to take a sample. The true future likely lies in combining paleoproteomics with other fields, like the study of ancient DNA and ancient metabolites. By layering these different molecular stories on top of each other, we can build a picture of the past that is more complete and vibrant than ever before. We are living in a new golden age of archaeology, an age where the stories of our ancestors are no longer just inferred from stones and bones, but are being whispered to us directly from the molecules of life itself. The secrets of the deep past are being unlocked, one protein at a time, and the journey of discovery has only just begun.